Hi, good afternoon and good morning, and thank you so much for the opportunity to talk to you today about lessons learned for future TB drug development, a review of rifapentine. So I remember several years back when Denny Mitchison made a bet with me that rifapentine would not be a better drug for TB treatment shortening than rifampicin, and we bet $5, a large sum, on this bet. And so I will leave you at the end of this talk to determine whether or not I won the bet with our well-respected late Dr. Mitchison. So the story to rifapentine, I think everyone has seen the recent New England Journal of Medicine article describing a 4-month rifapentine and moxifloxacin-based regimen that successfully treated patients with tuberculosis in just 4 months. In this trial, rifapentine plus moxifloxacin regimen achieved non-inferiority for efficacy in all analyses, while the rifapentine regimen without moxifloxacin substitution did not achieve non-inferiority for efficacy in any analysis. So how did we get here? And you can see rifapentine, the hand is rising up out of the grave. This was a drug that was developed in 1998 and really left on the shelf for a while when use of it and intermittently for TB therapy was really just not as good as using standard of care. So there are several people responsible for the revival of rifapentine. Among these are some clinical researchers, among them Dick Chason, but these 3 gentlemen were responsible for assessing this drug in preclinical models. And you'll see here in Rosenthal on the left, Eric Nurnberger and Jacques Grosset, and you may not see him because he's blocked by my picture, but he is saying, "Oh, this is not so stupid." He became very interested in rifapentine, as did the laboratory group here at Johns Hopkins. And so they showed back in the day that using rifapentine at 10 milligrams per kilogram achieved cure in 12 weeks of therapy in similar fashion to rifampin at 4 times the dose at 40 milligrams per kilogram. At the time, and even preceding these results, was some evidence that P here, rifapentine, with moxifloxacin had good activity for treatment of tuberculosis, provided it was given in daily regimens. At the time, there was some interest in intermittent regimens, but it turns out that using this drug daily was promising. So the lessons we learned here, and these are going to pop up throughout our slides, are that the BALB/c mouse model is excellent for identifying drug combinations that merit clinical evaluation, but we should just keep in mind that dose exposure and exposure response will be very different in mice than in humans. And so these preclinical experiments led to Tuberculosis Trials Consortium Study 29, in which the efficacy of rifampicin, given at 10 milligrams per kilogram, was compared to this new drug, rifapentine, at 10 milligrams per kilogram also. It was expected that rifapentine would really blow rifampicin out of the water. Much longer half-life, much lower minimum inhibitory concentration against M. tuberculosis. And yet, in the end, you can see here that on liquid culture, the culture conversion at 8 weeks was very similar with rifampin and for rifapentine, and the same thing was shown on solid media. And so this was somewhat disappointing at the time, and it was unclear really what these results meant. Does that mean that rifapentine is not better than rifampin? Was it given at the right dose? Or is it that our 8-week culture data simply were not telling the story, and that really it was going to be better and should be given at this 10 milligram per kilogram dose? And so this is the main lesson here is that dose finding has a role in phase 2 studies of multi-drug combinations. And if there's any sort of uncertainty about the dose or the combination that should be tested, using more arms, doing more arms up front is a good investment. And so this is just to show you some subsequent mouse study data. And you can see on the left, this is the BALB/C mouse. And if you use a rifampin-based regimen, that the colony-forming units are reduced over a period of time of treatment, in this case 4 weeks. And then with rifapentine, you get much better treatment response here. However, in the more complex Kramnik mice, you can see that yes, rifapentine on average performs better than rifampicin, but what are these two guys right here? Who are these mice? And so we know that with more complex lung disease, that perhaps some of these mice were not doing quite as well as were seen in the BALB/C experiments. And so these subsequent relapse experiments suggested that you could get about 2-month treatment shortening with rifapentine in the BALB/C, but somewhat less significant treatment shortening with rifapentine in the Kramnik mouse models. And so the lessons here are that Kramnik mouse models and other animal models that have more complex lung pathology can yield critically important information about variability in treatment response and drug exposures at the site of disease. And so thinking forward, the next idea was to test rifapentine at different doses to see if higher doses would give better treatment response and reduce the time to culture conversion. But first, we had to see these higher doses had never been tested in humans, and so first we designed study 29B, which looked at, which was a rifapentine dose escalation trial, starting with 5 milligrams per kilogram, going up to a maximum of 20 milligrams per kilogram among healthy volunteers, individuals that did not have TB disease. And you can see here that there were several dose-limiting toxicities. We had set up the trial so that if there were 2 dose-limiting toxicities defined as grade 3 or higher adverse events in any dosing cohort, that the maximal tolerated dose would be achieved. But you can see that in a 10 milligram per kilogram arm, after 7 doses, 1 person had grade 3 liver enzyme elevations. Another person, after rifapentine 15 mg per kg, after 14 doses, had lymphopenia, fever, dizziness, and liver function testing increases. Had there been 2 people having these side effects, then perhaps this drug would have been stopped in its development at the lower doses, which would have been quite unfortunate. So just something to learn about the design of dose escalation studies. Certainly adverse effects are different in people who are healthy volunteers than in people with TB. And in the course of study 29B, we looked at midazolam, which is a CYP3A probe, and found that the induction of metabolizing enzymes was as potent or more potent with rifapentine than with rifampicin. And so the lessons learned here are that the safety of drugs may be different in healthy volunteers than in patients with TB disease. We still don't really understand this. And that rifapentine, unfortunately, is not a weaker inducer of metabolizing enzymes than rifampicin. And so we need to keep that in mind as rifapentine progressed in its development. So then looking at 29B data, Radha Savage came into the picture in tuberculosis at around this time, and she showed us that rifapentine clearance is not affected by body weight, that rifapentine bioavailability, unlike rifampicin, decreases with dose. And so this was important because we're used to using mg per kg dosing in tuberculosis, but certainly it doesn't make sense to do that if weight is not impacting clearance in a meaningful way. Additionally, in study 5311, which was an ATTG trial, we found in healthy volunteers that these higher doses of rifapentine, given either in divided doses or with an egg to try to increase the exposures, led to toxicity in many cases. So neutropenia, rifamycin hypersensitivity syndrome, and we still don't really understand if that was something related to tolerability in healthy volunteers without TB that is different in patients with TB, or if these doses simply were too high and these exposures were too high and not well tolerated. We do also recognize here that mg per kg dosing is often not appropriate and really complicates treatment unnecessarily. So from TBTC Study 29X, this was the dose-ranging trial of rifapentine given after the healthy volunteer study showed that a maximal tolerated dose of 20 mg per kg was possible. Then the TBTC proceeded to Study 29X. And in this study, patients were randomized to rifapentine 10 mg per kg, 15 mg per kg, or 20 milligrams per kilogram, and there was a rifampicin control arm. And you can see here that all the doses of rifapentine seemed to do better than rifampicin, which was very encouraging. However, you couldn't really see a dose effect here. Was 10 and different from 15, different from 20? And I think that this is largely in part because that the dosing was in milligrams per kilogram, and yet clearance did not impact, was not impacted by weight. So people in different dosing arms actually had, in some cases, similar exposures. But when you look at the actual dose in milligrams, or you look at exposure response, then now we're starting to see some differences in treatment response by exposure or by dose in milligrams. And so the lesson here is that dose effects may be hard to see, particularly if you're doing mg per kg dosing, but it depends really on how you look, and that exposure response evaluations are an essential part of middle drug development, and they should be considered as such rather than as sub-studies. And so this is just showing the PKPD relationship for rifapentine. Here you see the area under the curve and the effect, and the maximal effect here is 4 weeks treatment shortening for culture conversion, and that maximal effect is achieved. Here's the steep part, or here's where the curve starts to flatten out when you get to a dose of about 1,200 milligrams, and this was the dose that was selected for the phase 3 trial. Lessons here are that quantitative pharmacology is essential to drug and regimen development, and that pharmacometricians really help us to see what we otherwise could not with our own eyes, and should be on protocol teams from their inception, as they're invaluable. And so from that same study, what became apparent is that there were patients with cavitary lung disease. So even though they had high drug exposures, that if they had large cavities on chest x-ray, that the percent of patients that had negative cultures by 8 weeks of therapy, even with high exposures to rifapentine, that culture conversion percentage was low. And so we started to see here that there were patients with cavitary lung disease that potentially would not benefit from rifapentine. Also in this, just starting to recognize who are the hard to treat patients, the patients that need to be cured for a phase 3 trial to be successful, and at least for rifapentine, and given in the combination with isoniazid, pyrazinamide, and ethambutol, those patients are people who have cavitary lung disease and high bacterial burden. And so for phase 3 success, we have to focus on these patients. And the hard to treat patients are often going to be different depending on the regimen, which we'll see in a minute. And so this was a really nice study done by Delan Rifat in the laboratory, looking at penetration of rifapentine versus rifampicin in a rabbit model of pulmonary disease that she developed. And you can see here that for rifampin, when you give a single dose, you don't have much drug exposure in these large lesions, but that with multiple dosing, that you get good penetration of rifampin into the lung lesions of these rabbits. However, with rifapentine, you don't see much drug concentration here coming into the lesion with single dose, and similarly, after multiple doses, there simply is not much rifapentine in these lung lesions. And so these drugs have different characteristics in terms of penetration into lesions. So to be successful, and I think this is one of the main lessons from the rifapentine trajectory, is that to be successful, drugs have to be present at the site of disease at sufficient concentrations to kill the bacteria, and that information about drug penetration into lung lesions should really be built into translational models and used in drug dose and regimen selection. And so following from the rabbit studies, Radha's group did some modeling and determined that drug concentrations in the cavity are about 25% of concentrations in the plasma. This is different than rifampin, which achieves similar concentrations in caesium as in plasma. And so she went further to determine what the target concentrations were, and it's 207 nanograms. The number is not important, but to achieve that concentration at the site of disease in individuals who have large pulmonary cavities, a dose of about 1,800 milligrams would be needed, and so a 1,200 milligram dose would not achieve that in many patients who have advanced pulmonary disease. And I should say that this target is specific for rifapentine when given in the context of that 4-drug regimen. And so this is just a teaser, but at the same time, the groups were doing studies of fluoroquinolone and showing that the activity within lesions as well as penetration of moxifloxacin both into rabbit lesions and also into human lungs among people who have advanced TB disease was very high. And so then came TBTC Study 31, and you can see here, and everyone is very familiar with these regimens, that people received either a rifapentine substituted for rifampin and shortened to 4-month therapy versus rifapentine substitution and also moxifloxacin subbing in for ethambutol, but also given for throughout the course of a 4-month treatment regimen. There were 34 clinical research sites in 13 countries on 4 continents. This was a really huge endeavor. Many investigators, many teams contributing to this. You can see the TBTC sites in green, the ACTG sites in red, and just with cross-network collaboration, involvement of well-established sites, and strong QA/QC, especially microbiology, great things can be achieved. This was a wonderful partnership. And so here are the primary efficacy results, and I won't belabor these are in the primary paper, but just to say that the moxifloxacin plus rifapentine regimen met all non-inferiority criteria in efficacy for all analyses, including among subgroups. And you can see here the NI margin at 6%, and all of these 95% confidence intervals fell well within that margin. And then you can see here that with the rifapentine regimen that none of the regimens met non-inferiority criteria. And so just looking at the unfavorable outcomes, I just thought that this was kind of interesting to share if you dive into the details a little bit, that of those with unfavorable outcomes in the control group here, only 34% of them were TB related, whereas in the rifapentine arm, 70% were TB related, and in the moxifloxacin rifapentine arm, about 51% were TB related. This is just to point out that there are a lot of things that might lead to an unfavorable outcome that are not related to tuberculosis, and in the context of a large trial, these tend to be similar across arms, and only with the significant sample size can you see the TB-related outcomes that you really care about. And here you can see TB-related unfavorable outcomes were much more common in the rifapentine arm than in the other arms. And so some lessons here, if just looking at patients who previously had been defined as high risk by previous studies, so people who had large pulmonary cavities or large bacterial burden, that with the rifapentine arm, those patients that were in the high risk category did not do well enough with that regimen, whereas those in the low risk category did. But when you have a really strong regimen with the drugs dosed at good levels, that the high risk patients are actually no longer high risk, that the outcomes were very similar among those that would be categorized as low, moderate, and high risk. And so lessons here are that with a good regimen given at effective doses, high risk patients are no longer high risk. And I just kind of wonder, with the right planning and the sample size, and pre-specified analyses, could the rifapentine-based regimen without moxifloxacin been registered as a 4-month regimen for low-risk patients? And so just to show you, these are the primary and secondary safety outcomes, and grade 3 to 5 adverse events incidence was very similar across the arm. Among individuals that had HIV, there were relatively few patients, about 214 randomized across the 3 arms. But I just wanted to point out that usually we think of HIV as being a risk factor for doing less well with TB treatment. But here, in the overall patient population, there were about 15% that had unfavorable outcomes, and that was very similar among patients who had HIV. And the same thing was not true, actually, among patients getting rifapentine. They had higher rates of unsuccessful among patients that had HIV than in the study in general. And the same thing was true with standard of care with HRZE. So just to show that moxifloxacin and rifapentine-based regimen was not only excellent among patients with HIV, but made HIV now no longer a risk factor for unsuccessful treatment outcome. And so the lessons here are, one, is just that the drug interactions had not really been worked out prior to the phase 3, and so there was a stepwise approach to including people living with HIV, so the number of people living with HIV in the trial was relatively low. So with some more advanced planning, perhaps that would have been different, and that with a potent regimen, HIV is no longer a risk factor for unfavorable treatment outcomes, which is a nice thing. In terms of emerging drug resistance, there was really very little acquired. So just the main lesson here is that acquired drug resistance risk is low with daily rifapentine-based regimens. And so overall, the lessons learned, well, there were many, and I hope we'll have some discussion and people can point out others that I didn't talk about today. But I think the number one thing is that translational and clinical pharmacology are central to TB drug and regimen development and should be considered as such. These are not sub-studies. These are really critical central components. But the BALB/c mouse model is very helpful in identifying promising regimens and is a useful tool, but should be used in conjunction with other preclinical models for decision making and really understanding which drugs have the best promise to go forward into clinical testing. Weight-based dosing is a thing in TB, but it really should only be employed when weight has a significant impact on clearance. Otherwise, it complicates therapy. And if you think ahead to fixed dose combinations, having a flat dose really does help with that, rather than multiple different potential doses. Also, I would say we should consider evaluating drug interactions with antiretrovirals that are commonly used in the field early in TB drug development, so that people living with HIV can join the late phase trials, and all of that drug interaction business has been really sorted out ahead of time. Safety and tolerability may be different in healthy volunteers than in patients with tuberculosis. This is the second main point. I would say drug penetration into complex lung lesions is an important aspect to understand in TB regimen development. Unfavorable outcomes in TB trials are only related to TB about half the time. If there's uncertainty about the right dose, and there often is, or the right companion drugs, so think back to 29X, think also to the phase 3 trial, run the extra arm, do the dose finding in phase 2, add the extra arm in phase 3 to make sure it's active enough. It will really be worth the investment. Pharmacometricians or pharmacomagicians are essential and should be included on protocol teams from the beginning. Large successful trials begin with strong partnerships, high quality sites, and good quality assurance and quality control of the microbiology. Certainly the ACTG TBTC partnership here was really fantastic. And then lastly, implementation does not necessarily follow from successful results and needs attention and advocacy. I just want to thank the wonderful colleagues I've had over the years at the Center for TB Research. There are many, but I'm just listing Dick Chason, Eric Narberger, and Jacques Grosset, who had the foresight to think that rifapentine was a drug that would be successful. Susan Dorman, who introduced me to rifapentine and really TB back when I was a fellow, and then across the world, the broad groups of individuals who were parts of Study 31 and ACTG 5349. Thank you very much, and I look forward to your questions.